welcome to all the audience. I hope since you just now had a very sumptuous lunch, I hope you guys would not doze off. So I have two responsibilities. One is to deliver the talk and the other is to keep you from dozing off. Okay, so now the topic that has been given to me is biotechnological breakthrough or breakthroughs in commercial crops enabling future farming. So I thought before going into this subject right away head on, there is no harm in having a retrospective of agriculture and the breakthroughs that happened in the past 200 years or so. I'm sure you all know that humankind has domesticated crops about 10 or 15,000 years ago in Mesopotamia, where human beings have domesticated wheat and barley, and a little bit after some time, humankind has domesticated maize in Mexico and nearby places. So we had an interregnum of so many centuries during which humans have selectively chosen the best performing varieties or variants or mutants suitable for his or her taste, palatability, storability, and so on. But the real breakthrough in agriculture and crop production came in 1865 or 19th century after the discovery of loss of inheritance or loss of genetics by Gregor Mendel. That was when the scientific plant breeding started. So we have a glimpse of all these revolutionary events and the people responsible for that a little later. But as you can see later, mutation breeding came up with the discovery of nuclear energy, nuclear radiation, then anther culture in our own backyard in Delhi. Anther culture has been done by Shipra Guha and Satish Maheshwari that gave rise to development of dihaploids, which are very, very important and essential in current plant breeding programs, and then genetic transformation in 1980s, followed by molecular breeding or marker-assisted selection, and people started employing molecular techniques like RAPD, RFLP, AFLP, microsatellites, and so on and so forth, going up to single gene-based molecular markers aided by genomics, bioinformatics, and so on. Then genome editing, the latest breakthrough, which I will spend a little bit more time on that. So let us recollect the real heroes of plant breeding. You see here, in 1950s, Orville Vogel has crossed Norin 10 and Brevor 14. Norin 10, please keep in mind, it has come from Japan, a semi-dwarf variety of wheat from Japan, which has been crossed by Vogel with American and Mexican wheats. And whatever material he has developed has been later utilized by Norman Borlaug in cement. And that's what gave us dwarf and semi-dwarf Mexican wheats that formed the substratum or the foundation for revolution in wheat, that is the Green Revolution. Very similarly, you can see in Iri, Philippines, Henry Beechel has crossed Peta, an Indonesian variety, an elite variety, with DG Uji, or DGWG. DG is the gene, the local name. And this was accessed from Taiwan. So an Indonesian variety crossed with a Taiwanese variety gave rise to short rices. And IR-8 was the primary short rice variety developed, popularized by Beecher and his successor, Gurdev Kush. And that has given rise a green revolution in rice in India. So we thank them. Mutation breeding, I just now mentioned after the discovery of nuclear energy, radiation, and so on and so forth, the mutation breeding has taken off. But before that, Louis Stadler has exposed seeds to X radiation, working in the University of Minnesota in 1928. And later, mutagenic agents like gamma radiation, UV radiation, chemical mutagens like EMS, etc., have given rise to something like 3,200 varieties between the period 1930 and 2014, which have revolutionized the production of several crops, cutting across all kinds of crops. And this is the haploid production which I have mentioned just now earlier. Shipra Guha, a graduate student of Panchanan Maheshwari, working as a postdoc with Panchanan Maheshwari's son, Satish Maheshwari, inadvertently, accidentally, found some anthers of Datura on tissue culture, discarded, and she has noticed some small plantlets coming out of Datura anthers. And that was the basis of haploid production, that is production of plantlets from pollen grains or haploids. And you know that dihaploids are occupying a very, very important place in current plant breeding programs. Molecular breeding, I am sure everybody knows about this, everybody heard about it, some of you are probably doing that, had its basis in 1923, based on morphological markers, followed by biochemical markers, followed by isoenzyme analysis based markers. Carol Sachs and later plant breeders have started making marker-based selection of varieties that formed the foundation for molecular breeding. But serious efforts that happened in 1980s, early 1980s, in Cornell University by Stephen Tanksley, who has made the first linkage map in tomato. And that actually resulted in a flowering of marker-assisted selection technology in all crops, important crops like rice, wheat, soybean, and so on and so forth. And we come to genetic engineering now, that is the 1980s, early 1980s, and the foundation was made by molecular plant pathologists investigating the crown gall disease, the pathogenic bacterium, that is Agrobacterium tumefaciens, the basis for crown gall formation. And this was pioneered by three great scientists, Mary Dell Chilton in Washington State University, Mark Van Montagu in University of Ghent, Belgium, and Joseph Schell in Max Planck Institute for Plant Breeding in Cologne. So these three scientists have dissected the mechanism of action of Agrobacterium tumefaciens induced crown gall disease, and they found that the basis for this disease is transfer of a discrete fragment of DNA from a circular plasmid containing in Agrobacterium tumefaciens to the plant cells that results in imbalance in auxin and cytokinin production, resulting in tumors, which they have exploited to make disarmed plasmids, removing unnecessary genes, and then incorporating desirable genes like, let us say, antibiotic production. And for the first time, A transgenic tobacco plant was developed in 1983 by these three groups independently almost at the same time. Papers were published in Nature and Science almost at the same time. That was the beginning of this great revolution, a great breakthrough. That is GM crops, genetically modified crops or genetically engineered crops or transgenic crops, whatever it is. The current status is, as you can see here, there are more than half a dozen non-food crops from cotton to petunia, flower crops, and more than 22 food crops, ranging from maize to golden rice. Golden rice also has been recently approved in August, September in Philippines and Australia. As you can see, most of the major food crops, cereal crops and other fruit, vegetable crops have been genetically modified and which are commercially available in 29 countries, cultivated in 29 countries, and 43 countries import GM crops, foods and products derived thereof. So much so, in 2022, GM crops have been cultivated in 190 million hectares in 29 countries. 29 plus 43, that many countries or that many citizens have been consuming GM foods and the products thereof. And the major traits are enumerated here. And the economic and environmental benefits are well documented in a period of 10 years or more than 10 years from 96 to 2018. Crop production has increased by value $225 billion. GM crops provided a better environment by saving 776 million kilograms of pesticides, reduced CO2 emissions in 1 year 2016, equivalent to removing 16 million cars off the roads. Conserved biodiversity by saving 231 million hectares of land by saving forests from being degraded or deforested. And the most important economic benefit is helping alleviate poverty for 17 million small and marginal farmers who are some of the poorest of the poor people in the world. And you know the benefits of BT cotton. I would not dwell too much on this because of lack of time. Then we go to genome editing. That is the latest breakthrough in agriculture. Starting from plant breeding, we have come to 21st century, that is 2019, alteration of the genetic material of a living organism, because this cuts across plants, animals, mammals, human beings, so on and so forth. By inserting, replacing, or deleting a DNA sequence, typically with the aim of improving some characteristic of a crop or a farm animal or any other organism, or genetic disorder correction or whatever. That is genome editing. So this genome editing gives different opportunities. As I have mentioned in the previous slides, you can delete the gene, you can modify the gene, you can alter the gene, and so on and so forth, by depending upon the naturally available mechanisms in the cell genetic machinery. For example, if you cut DNA, a double-stranded DNA, you can delete part of the DNA or you can insert part of the DNA in that cut. And this is what is called non-homologous end joining. Here, the DNA automatically heals itself by the help of naturally occurring DNA repair enzymes. The other approach is homology-directed repair. That's where you are going to cut the DNA and you are going to give an external DNA template, a homologous sequence of DNA. So based on the homology between the native DNA and the externally given DNA, there will be an exchange of DNA strand, and automatically the DNA is repaired. So you have a deletion or a modification or an insertion even by homology directed repair. So these are the two approaches in genome editing. And genome editing technologies have been around for more than 15, 18 years. In fact, CRISPR-Cas9, the one which I mentioned earlier, is the latest. Earlier, scientists have been doing this by depending upon zinc finger nucleases, later on talons. This is about 10 or 12 years old technology, transcription activator like effector nucleases. These two genome editing technologies have now become almost redundant or moribund, replaced by the genome editing technology based on CRISPRs, clustered, regularly interspaced, short palindromic repeat. I never remember this. So I have to read this. Every time I have to read that. So people also, even people who have discovered all these things, they put CRISPR to that. CRISPR, simple name. So you simply call it CRISPR. So these are the three genome editing technologies, and the bottom one is the one which we are now talking about. And for this discovery, two ladies, great lady scientists, Jennifer Doudna, based in University of California, Berkeley, and Emmanuel Charpentier, working in Max Planck Institute for Science of Pathogen, Berlin. They shared the Nobel Prize in Chemistry in 2020 for discovering genome editing by this CRISPR-associated nuclease technology. How does it work? Because this particular mechanism actually is again a naturally occurring mechanism present in microorganisms. Recollect the Agrobacterium tumefaciens, a microorganism, a plant pathogenic microorganism that gave rise to the most powerful genetic engineering technology. Here again we are depending upon a bacterium which has a defense mechanism to ward off the invading viruses, bacteriophages and other viruses. What bacteria cleverly do is, as you can see here, a virus invades the cell, bacterial cell. Then the bacterial cell captures a small part of the viral genome and integrates it into CRISPRs. As you can see here in light green color square, that is the viral genome part getting integrated into CRISPR gene. Now, the bacterium has another mechanism which will guide this particular CRISPR towards the other invading virus. Then the original DNA that has been taken in serves as the template, and there is a double-stranded DNA cutting enzyme, CRISPR associated nuclease. So at that particular place where you are going to have that homology between the viral genome and the CRISPR captured genome, the enzyme cuts like a scissors. And once the DNA of the virus is cut, the virus is unable to multiply itself. So this happens hundreds and thousands of lakhs of millions of times. So the microorganism, or the bacterium in this case, protects itself from the virus. So this mechanism has been exploited by these two clever scientists to develop an artificial CRISPR associated nuclease. And here you can see little complicated for beginners or who are not molecular biologists or molecular virologists. So here you have a single guide RNA followed by CRISPR associated protein 9, then a transactivating CRISPR RNA and CRISPR RNA. These four are the components of this whole mechanism, and the cartoon is given at the bottom how this happens and how the DNA double strand is cut, how the guide RNA takes the CRISPR there, and how the gene can be edited. So this we can do in plants also. In fact, this has been first demonstrated in microorganisms and later in small mammals like mice and so on and so forth. But plant molecular biologists have immediately caught upon this knowledge and started exploiting that. So you have to make special vectors by name CRISPR-Cas vectors, in which you have to have a small stretch of DNA, that is the template DNA, where you want to do the editing in the plant cells, and then by using these synthesized vectors, you again depend upon Agrobacterium tumefaciens to insert it into the plant cell. Nowadays you have technologies that bypass Agrobacterium also. You can also do this by particle bombardment or gene gun, but whatever it is here, this particular picture shows you Agrobacterium tumefaciens at the top and gene gun or particle bombardment at the bottom. You are going to bombard or infect the plant cells, the plant of your choice, and from that you regenerate in tissue culture shoots, hundreds and thousands of shoots, and then you screen the shoots. Even if you develop a handful of shoots or 10, 20 shoots, it's enough. But each and every shoot, every plant has to be analyzed by molecular techniques like PCR or even genome sequence. And after finding out the gene-edited plantlets, then you are going to subject it to testing in your glass houses or laboratory culture rooms, whether it is for resistance to a fungus, as you can see here. It is actually a rice plant which has been challenged with a bacterium, or I think it is fungus. And then the resistant plants are carefully grown and then taken to glass houses and then fields. Fortunately, the government of India recently has given some kind of relief for those scientists who are working in genome editing. Unlike in genetic engineering, for two categories of genome editing, there is no regulatory mandatory requirement. If you inform your institute biosafety committee, give all the details of your experiments, you prove that the editing is successful and there is no extra unnecessary DNA coming from Agrobacterium or through particle bombardment vectors. If that data is enough and you can go directly into the field, you can do field testing and you can go for commercial release of that. Only two categories out of 4 categories. So here you see, after 2012 or 2013, the plant scientists have taken to genome editing in a big way. For example, in one crop rice, this data gives you examples only up to 2017. But in the past 3 to 4 years, there is an explosion of knowledge and also genome edited plants and species. As you can see in example rice, you have grain weight, early maturity, photoperiod, grain number, panicle architecture, grain size, tiller number, drought tolerance. So many characters have been altered. Again, mind you, this genome editing is only possible if you have a gene already present in the organism that needs to be modified or silenced or whatever. If there is no gene, then you have to go back to transgenic crops or genetic engineering, where you are going to add a foreign gene from outside. And in horticultural crops, you can see again a variety of traits or characters that have been edited, gene edited in horticultural crops, like tomato, carrot, potato, sweet potato, so on and so forth. So much so, in 2021, Japan has approved, or rather allowed, genome edited tomato into the market. That is a company by name Sanatec has commercialized this tomato. This expresses very high levels of GABA or gamma aminobutyric acid. It is developed through CRISPR-Cas9 technology. GABA is an amino acid that aids in relaxation, lowers your hypertension or blood pressure. It contains 4 to 5 times more amount of GABA than a regular tomato. So this is one of the earliest examples of genome-edited crops that are available commercially now. And the opportunities when you come to, let us say, the NIRCA, are the crops that are being made as mandate crops of NIRCA. In addition to tobacco, you have chilies, turmeric, and Withania. Withania is ashwagandha, and I am very sorry that I could not include castor in this. I came to know very recently that castor also has been added to this. But you can see the opportunities crop by crop, species by species. In tobacco, molecular breeding, of course, it is not a recent breakthrough. It's a relatively older breakthrough, where you can depend on molecular breeding for disease and pest resistance, especially when you are talking of nematodes, the nematode problem. You can depend upon marker-assisted selection. And where you are unable to do that, for example, in disease resistance, you have to take a resort to CRISPR-Cas9 technology. Same way in chilies, as many of you know, it has very, very serious insect problems and also disease problems, which can be manipulated or managed by molecular breeding, and genome editing especially can come to your rescue in incorporating disease resistance. Same way in turmeric, disease resistance is one thing. The other thing is curcumin content. You can enhance the curcumin content by manipulating the pathway of curcumin biosynthesis by genome editing. You find out the bottleneck enzymes and the respective genes, do the editing, and thereby enhance the curcumin content. And this is just one of the opportunities. I haven't, in fact, listed all the available opportunities. And in ashwagandha, in fact, you all know that the medicinal properties are because of alkaloids like withanolides. They are called withanolides. And withanolide content also can be manipulated, enhanced by working out the metabolic pathway of withanolide biosynthesis. So this is one opportunity. And during the presentation of Dr. Madhav, I have come to know that CTRI or NIRCA has already started working on genome editing with respect to solanisol content by identifying two genes, that is DXS and DXR. So where you can do genome editing and enhance the content of solanisol. It's a very, very expensive chemical molecule, and you can earn a lot of revenue for the institute or for the organization. Same way, genome editing will come to your help, rescue in lowering the content of nitrosamines, which are very, very, very carcinogenic compounds, and they have already identified CYP gene responsible for the biosynthesis of nitrosamines. So I wish all the best in these efforts of genome editing in tobacco by CTRI or NIRCA. I'm sure if you are handling a few nucleotides or one or two base pairs, you can right away go into the field once you identify the edited events by molecular as well as agronomic analysis. So I wish all the best, and this is what I thought I would highlight. Although this is a very, very short list of opportunities that are available, but opportunities are many. As they said, when genetic engineering first came, they said sky is the limit. In fact, the limit is human imagination for genetic engineering. The same thing applies for genome editing also. Human imagination is the limit for this. And in tobacco, of course, this has already been presented by Dr. Madhav. There are so many phytochemicals, so many useful metabolites. So all these things actually will give you lots and lots of revenue. And also it will help you removing the stigma of tobacco being considered as some kind of pariah crop or whatever. So you can make this particular crop commercially viable, commercially productive, and make lots and lots of money for the country. And these are antioxidants, anti-parasitic, antifungal, antibacterial molecules. So many kinds of molecules are present in tobacco, which you can manipulate by genome editing as well as by other techniques. I think that's where I have come to the end of my talk. Thank you very much.
Thank you, sir. I mean, you elaborately talked from evolution of plant breeding to genome editing. We will take up the
questions.
yeah later, sir.
Okay, fine.
Yeah.
Thank you.
Now I request